Hello there, genetics students. All right, so here we are with chapter 3. Again, another long one, but let's jump right in and hopefully let's try again for 3 videos. So for a long time, people believed that traits were passed on to offsprings in a number of different ways. Very early on, Hippocrates, one of the great philosophers, suggested that seeds produced in the body were collected and then transmitted to the offspring at the time that the individual was conceived. The problem with that is that there aren't individual seeds for all the different body parts. After that, the idea of preformationism came up, and this came up because in the early advent of the microscope, people believed that they could see literally little humans inside of sperm or an ova. So if you thought that you saw a little person, which they would call a homunculus, inside of a sperm, that person was considered what they would call a spermist. The ovists thought they would see the person in the ovum. And so, that was one idea. So the idea was just that one of the gametes carried the individual, the other gamete nourished it. And that's not a bad idea. And in fact, to some degree, that's kind of what happens, but not entirely. And then there was the idea of blending, where factors that control your heredity or the individual's heredity, those factors were kind of malleable. They were able to be blended like colors on a color wheel. And so that after generation, all these traits would come together and blend into one individual. The problem with that particular idea was that once things were blended, you could never separate them apart. And we know that there are obvious traits that are transmitted through generations. And we'll get into that as we work through this particular lecture. So starting in about the mid-1800s, a scientist named Gregor Mendel started working on how traits were transmitted from parent plants to their offspring. And one of the interesting things about Mendel was that Mendel himself was an individual that was very curious about how traits are transmitted, but moreover, Mendel used a new technique, statistics, to understand what was happening in the transmission of traits. And so as he was working, he did actual quantitative analysis on how traits are passed from one generation to the next. And this hadn't really been done very much before. Quantitative analysis now is commonplace in scientific research, but at the time, Mendel presented his work and showed data for his work, and people went, "Thanks, that's awesome," and they didn't really understand what it meant until the early 1900s. But what Mendel came up with out of his work were 2 laws. One of them that stated that you could predict what the phenotypes of the offspring would be in the next generation. And that prediction could be done in specific ratios as long as genes had an either or type of phenotype. So we're going to get into that. His work was not abandoned, but not understood very well until the 1900s, when people started looking at the transmission of traits in more complicated organisms, in vertebrates and so forth. So let's move on with what Mendel worked on. So Mendel worked with pea plants, and the reason why he worked with pea plants is because pea plants were small. You could grow them easily. Pea plants have flowers that have male and female structures, and we talked about this a little bit in the last lecture. The male structures can be removed so that the plant could be specifically pollinated with pollen from a different plant. So you could do specific crossbreeding or fertilization experiments. Plants could also self-fertilize so that you could look at what genes are present in the plant itself. And the variables that Mendel looked at were flower color, stem length, tall or short, how the pea pods looked, whether the pods were inflated and smooth or kind of lumpy, the height of the flower, the height of the plant, flower position, and so forth. And so in this picture, what we're looking at here is just an artist's rendition of a pea flower. So the sepals here are leaves that are adjacent to the petals. The sepals close over the petal before the flower emerges. So as the sepals peel back, the petals emerge. The keel is a central petal. The stigma is a part of the female reproductive structure. The stigma is the tip of the style. The ovary is where the ovules are housed. And then each individual branch over here are, and the male portion, are anthers, and at the tips of the anthers are where the pollen or the male gametes reside. And so what has to happen is that the pollen leaves the anthers and interacts with the stigma, and then the pollen tube extends down through the style for fertilization to occur. And so we saw this picture before. Here's a picture of a pollen grain sending a pollen tube with 2 sperm nuclei down into the ovary. And inside of the ovary, you have a central cell with 2 polar nuclei. That'll become important a little bit later. You have the ovule, which has the embryo sac inside of it, and then the actual eggs down here, which are haploid. Bear in mind that the 2 sperm nuclei here are also haploid. Sperm nuclei, ovum nuclei merge and become a diploid zygote. The micropyle down here on the bottom is this location just below the eggs. And so the pollen tube grows into the micropyle, goes all the way around down here. And the 2 sperm nuclei unites with the 2 polar nuclei of the central cell. Sometimes you have double fertilization. Don't worry about that right now. So here's some of the variants that Mendel looked at. Height, tall dwarf. Flower color, purple or white. Flower position, axial. So in other words, the flowers were on the side. Terminal, the flowers were at the very tip of the branches. The seed color, yellow or green. So that's your yellow peas and your green peas. Seed shape, round or wrinkled. Pod color, green or yellow. And then pod shape, here's the smooth or constricted or bumpy shape. And so Mendel did 2 types of crosses. He allowed plants to either self-fertilize. The pollen and the egg came from the same plant. So in other words, the pollen landed on the stigma of its own plant and fertilization occurred there. Or cross-pollination. When cross-pollination was done, what Mendel did was, let me come back to our little figure up here. Mendel would take a pair of scissors and cut off the anthers of the flower before pollen was produced, and then he would take pollen from a different flower on a paintbrush and paint it onto the stigma of a different plant. So he literally would transfer pollen from one plant to the next, and then he would cover the flower with a little burlap bag to ensure that further pollination from other plants didn't occur. Oh, I could have just gone down to this picture, so here you go. Here's the removal of the anthers and then pollen transfer, say from a white flower to a purple flower. After cross-pollination occurs, you have the development of the seeds, and each seed themselves is an individual outcome. The pea pod can be considered an individual outcome. The color of the seed is an outcome. And then once you grow the plants in the next growing season, this is what we're going to call the first generation offspring. So the parental generation, or the pea generation, would be the purple and the white. And then F1, or the first filial generation, are the kids, the offspring of the purple and white blending. Now, one of the things that Mendel wanted to make sure he knew was what are the genetic components of each of the parents that he's using in these hybridization or breeding experiments. And so what Mendel actually did for the first couple breeding seasons was he bred plants that were what were called true breeding. In other words, when the plant mates with itself or any other plant, you get the same result of offspring. So a purple plant mating with a purple plant is always going to give rise to another purple plant, or the purple plant self-fertilizing is always going to give you purple. So he started with true purple plants and true white flowering plants. I should say purple flowering plants. And so what that means is that the trait that he was looking at didn't change from generation to generation because of changes in the organism itself. Okay? When he mated these true breeding plants, what he noticed was that sometimes some of the offspring looked different. So when you look at matings and you look at just one, when you produce a mating and you look at just one character, excuse me, say like flower color, you call that a monohybrid cross. Mono for one, looking at one thing. Hybrid, you're breeding for one thing, and then the cross just means that you're crossbreeding 2 organisms. Now, when you're looking at 2 characters, let's say flower color and stem length, how tall the plant is, okay? That's a dihybrid cross. Di for looking at two characters, and then hybrid for the hybridization or the cross. And so in Mendel's experiments, he just kind of went along with, okay, he's trying to figure out what he was going to get from these hybridization experiments. He didn't have a hypothesis to explain what would happen when true breeding purple and true breeding white came together. So instead, he just used his quantitative analysis. He counted the number of offsprings in the F1 and F2 generations that contained certain traits to look at the odds that you would get that offspring potential. And so this was a mathematical and statistical analysis. And so what we call this now is an empirical approach. And so the empirical method allows you to count something and then deduce an empirical, or basically an analytical law of a process. And so the 7 characters that Mendel dealt with, each character had 2 variants or 2 traits, so like white or purple flower colors, or forms. Here, tall or dwarf. And so these first experiments that he did, he simply did monohybrid crosses. Now, what's interesting about Mendel was that he kind of thought about these crosses in a unique way. I know you're probably already familiar with how to use a Punnett square. The way Mendel thought of these experiments was more like flow charts, not like 4 squares and then the parental genotypes, which we'll get to in a few minutes. What he thought about was, okay, I have tall plants and short plants. And so the idea is that you have 2 components that will give you a tall plant and 2 components that would give you a dwarf. In the F1, you're going to have a blending of the 2. So when you plant those, you're going to have, oh, look at that, all tall plants. When you allow F1 to self-fertilize, when you look at what is inside of that particular genotype, you'll notice that 3/4 of the plants end up tall and 1 is dwarf. And so what this tells you is that short is in there somewhere. You just didn't see it in the F1. Here's some actual data from that first experiment. And what he saw was that a tall plant and a dwarf stem, it was a 2.84 to 1 ratio. Now, we'll get to this type of statistics a little bit later on. I just want to leave that in there for you. So, what did he take away from this experiment? For all the characters that he studied, the F1 generation showed only one of the 2 parental traits, and it always showed the most dominant trait, which we're going to get to in a little bit. The F2 showed both traits. So if you had a tall plant and a short plant, okay, and 3/4 were tall and 1/4 was short, the short was always in there. You just couldn't see it in the F1 generation or the first filial generation. And so these results refuted the blending mechanism of heredity, because in the F2 generation, in the grandkids, you saw one of the parental components. So in other words, things don't get mashed together and mixed over time. Things come together, you see one or another, and then they can be separated back out. Now, one thing that you need to be aware of is the level that Mendel was looking at was simply flower color, height. There are numerous factors that can influence these particular traits, which we'll get to later on. These particular traits are either or traits, all right? So the trait is going to be dominant or recessive. So what Mendel postulated from this experiment was, for any given character in this particular experiment, pea plants contain 2 heredity factors, one from each parent. The 2 factors can be identical or different, and of those, the one that you see is the dominant. The dominant is the one whose effect can always be seen. The recessive is not expressed. All right. Now, the recessive, as it turns out, really is expressed, but you can't see it. So during gamete formation, these 2 traits are split apart from each other. So in other words, you have a cell that has big T, little t, but when gametes are formed, big T and little t are separated from each other. And from this, Mendel came up with what he called the law of segregation. Traits are separated from one another at the formation of gametes. All right, we're going to stop here, take a break, and pick up with the next experiment in just a minute.